class schedule and talk a little bit about stem cells to kind of help set the stage and give us a break from talking about the march through the germ layers and what they become. So, topic here for embryonic stem cells and these induced pluripotent cells, and then we'll have an in-class exercise to do here in a little bit too. Okay, so stem cells in general, what are they? And stem cells are really important for the body during development as the embryo is forming and growing and shaping, but then also in the adult organism to continue to maintain an adequate source of cells for all of our different tissue types. And there are a very special population of cells that are called stem cells, and they all share a couple of basic properties. First of all, they have the distinguishing characteristic of long-term self-renewal. And that means that these cells can go through mitoses after mitoses after mitoses, and they just keep on going, and can generally produce daughter cells over and over and over again. And, you know, not every cell type in the body really does that. You know, you would think that there's nothing to prevent them, but many cell types, as they get more differentiated, really only go through a rather limited number of subsequent divisions. So for example, like epidermal cells, they're going to become your spinous cells, your granular cells, and then they'll go into forming those flat cells of the cuticle, and that's it for them. They don't divide after that. Or neurons. You know, initially there's a certain population, and then once you get to a fully differentiated neuron, you don't see those cells dividing a lot. But these stem cells do. And some of them are always reserved to be continuing as stem cells. So some of them will be daughter cells that are stem cells themselves, and then some of them will differentiate into mature cells with any of the different cell fates that you can think of. So another thing about stem cells is they're pluripotent, meaning that the types of cells that they differentiate to could be more than one thing, and that somewhat just depends on the stem cell itself. There are some cells that are very highly pluripotent and can give rise to all kinds of different daughter cells. And then there are some cells that are stem cells, but they really only give rise to one type of cell. So for example, the spermatogonial cells that we find in the testes, those are stem cells. They divide over and over again. They make literally thousands of sperm, but they really only make sperm, and that's it, not much else. So 3 types of cells that we'll talk about here are adult stem cells, embryonic stem cells, and then the induced pluripotent stem cells. So there are various different populations of adult stem cells around our body, and I think some of these we're well aware of and others are still a bit more mysterious. So for example, cells that have to proliferate a lot, like blood cells, spermatogonial cells, cells in the epidermis, those we're all aware of. There must be some source, even as adults, of stem cells that continue to proliferate. But then there's been a lot of excitement and research done over the past 10-15 years about cells in the bone marrow that might travel around the body and function as stem cells to contribute to different types of regenerative potential, at least at some level, even in the adult. So maybe there are some cells in the bone marrow that might eventually colonize the heart and function as a source of new cardiomyocytes, so a new differentiated heart cell. And there is some evidence to suggest that this can happen, but on the whole, these cells are much more elusive and harder to find, harder to track, and harder to study. So, and then some populations we maybe think are, maybe there doesn't exist an adult stem cell, or if so, they're very rare and few in number. Stem cells often have what's called a niche that supports their continued growth, survival, and proliferation. And what this would involve is a specialized environment where these stem cells reside and where they stay alive and where they continue to proliferate. So this is just a diagram I took from a recent review. And this green cell here in the middle, that's the undifferentiated stem cell. And you can see it's in contact with maybe certain structural features or physical features that surround that cell. It might be getting certain paracrine factors from nearby cells. It might be getting certain neural input. It could also be getting signals, humoral signals. This means from the blood or other body fluids. So for example, hormones might be being brought in to nourish that stem cell and keep it alive and dividing. So as long as the stem cell is here in this special niche environment, it will continue as a stem cell. But then some of the daughter cells are going to leave that, and then they'll be further away from the special environment, from the source of these paracrine factors, and at that point they could go ahead and differentiate to whatever type of downstream daughter cell they're going to make. So a niche is another thing that we need to know about this and understand it. We're really going to understand stem cells and where they come from. So here's an example. The hematopoietic stem cells are very proliferative. They're going to give rise to a lot of our different components of blood, but also some of the cells involved in immunity. So here's just a little cross-section through a bone. There might be some bone matrix, and then here's some of these osteoblast cells that are just making additional bone. And then over here on this side we have maybe some innervation from vascular tissue. And then hematic stem cell, HSC, hematopoietic stem cell. This is in initially a quiescent state or in a kind of a not very active state. But then once it comes in contact with the appropriate juxtacrine signals in the environment from other cells, endocrine signals coming from the vascular system, maybe paracrine signals coming from other cells in the region, the combination of these will then trigger that hematopoietic stem cell to be more in an activated situation where it can give rise to a variety of different downstream cells. Okay, so this may be one of our better studied examples of a stem cell niche here within the bone marrow, and we know the identity of some of these cues that can activate that hematopoietic stem cell. You could also think about stem cells in the context of evolutionary biology and how some animals differ from others in their ability to regenerate or not regenerate. So for example, the zebrafish, it turns out, adult zebrafish hearts, you can go in there and severely damage, up to half the heart. That thing can regenerate. And we don't know why, but this is a vertebrate, and this is definitely not something that humans can do or mice can do or any of those guys. So once again, zebrafish come through, and they will learn something from that species about how we might manipulate mammalian cells to regenerate. This is an example of a rodent that has some bottom teeth that, due to their life history, they're always gnawing and wearing down these teeth. So throughout the life of the organism, the teeth continue to grow. And so there are actually a source of stem cells down here that are continuing to divide, and they're going to contribute to the odontoblasts that make the dentin, which is the bulk of your tooth, and these amelioblasts, which make the enamel that coat the outside of the tooth. So these stem cells are present here in this location at the base of the tooth in the rodent, and they're busy making more teeth. And in other species, there are no stem cells active throughout the life of the organism here, and you do not get the continued growth of the teeth. So interesting to think about maybe what's different in this niche area between the rodent versus something that does not have continual tooth growth. Okay, stem cells have a lot of interest these days in the scientific community for a number of different reasons, and here are just some of the reasons why people find these interesting. One is that they are a way for us, a tool for us to try to understand how cells make these choices about cell fate. So, that's part and parcel understanding how development happens, and so if you can take what's initially a generic cell in a dish and then figure out what specific gene products or factors or conditions will stimulate it to go down a particular fate, say cardiac cell fate versus neuron, then that's a good way to learn about how these cells normally differentiate. Secondly, drug safety testing. So there is extensive testing of all of our cosmetics, our medication, things that we get exposed to, and much of this is done on animals. And it's probably necessary to do this, but it costs a lot of animal life, some animal suffering, and when you get down to it, the animal is not human anyway, so this could potentially be a complementary avenue where you could, say, pre-test products on stem cells to see if they're utterly toxic or if they survive or what kind of exposure the cells end up getting. And then finally, an area that's kind of growing momentum is the idea to use stem cells for therapeutic purposes. So if there's a human out there that has some kind of developmental disorder or a disease that's progressing, could we actually find and use a set of stem cells to correct the problem? And what would be some of the challenges or limitations of this? So one good thing about it is that you have pretty much an unlimited source of cells. You can always grow more stem cells in culture, and as long as you know how to differentiate them to the cell that you want, and as long as you know how to get them to the place that they need to be, you might have a really amazing tool to replace cells that have died due to disease. And also, if you have the ability to collect these initial cells from the same individual, put them in a petri dish, differentiate them as you like, then those cells won't have any immunology rejection issues when you put it back into the same person because they're already recognized as being self by the immune system. So that could also be a huge bonus for stem cell work. So could we use adult stem cells to try to pursue some of these therapies? And there has been interest in this, but here are some of the limitations. So we don't actually have an adult stem cell for every single tissue type. So for example, the cardiomyocyte precursor cell is a little bit elusive and hard to pin down, hard to isolate because there's just not very many of them and we're not really sure where they're coming from. And then also these cells might have already accumulated some mutations because these cells have been around in the organism for 30 years, 40 years, 50 years, and might have acquired some somatic mutations that would still be there. We still would need to manipulate these cells to get them to go to the final cell fate that we want. So, ethically it's not so much a problem to use these kind of cells, but just for very practical, logistical purposes, it's often difficult to find the appropriate number or possibility. Another option would be to use embryonic stem cells. And these are cells that are derived from a blastocyst inner cell mass. So when we talked about blastocysts, we pointed out this group of cells that were very highly pluripotent. And these were the cells that would give rise to every tissue that existed in the embryo itself. So, it's pretty easy to get blastocysts either by just fertilizing in the dish or collecting them from, say, mice that have been mated in a timed fashion. And then you can harvest out these inner cell mast cells and keep them happy and proliferate in the petri dish, and then apply different strategies to get them to differentiate to a tissue type of choice. And so there are a limited number of lines that are existing for human embryonic stem cells, and these might have arisen probably not from blastocysts, but from embryos that were legally aborted or other very early human tissue sources. And then once you get these lines, you could propagate them and use them for experiments to explore possibilities here. However, of course, to make these cell lines does involve some embryos and possibly the destruction of embryos, and they don't last forever. So even though they're embryonic stem cells, they tend to want to differentiate in culture, and so sometimes the lines lose some of their pluripotency or they start to differentiate. So they're not perfect. Just a little bit about how these are maintained. It's a bit tricky, so once they're harvested, they have to actually be grown on another layer of cells because they don't like contacting the petri dish directly. That tends to make them just differentiate or die. So it's a real struggle to try to keep these guys in a really highly pluripotent state so that you could use them for the experiments that you want. So people use these fibroblast feeder cells, which are just kind of layers of cells that have been irradiated so that they themselves don't tend to divide very much. And then the only cells that are growing and proliferating are the stem cells, and they're just in contact and touching with fibroblast cells, which is more compatible for them. And then as they get too many cells in a dish, you have to split these and passage them out to a new petri dish with new feeder cells. And so there's some work involved in trying to keep these cultures alive and growing and proliferating. Then there comes the challenge of what do we do with them? How do we get them to differentiate into the cell type of issue or of interest? And sometimes we know some of the factors that would do this, and sometimes we're not really that sure. So for example, you could start out with an ES cell, and if you expose it to a media containing retinoic acid, which is a paracrine factor, that can push those cells into more of a neural stem cell fate. And then from that, they give rise to actually functional neurons. So you might get them to this precursor state in the petri dish, then transfer them over to the organism, and in that place they can complete the process of growing and integrating and establishing themselves as functional neurons within the organism. Or here's a different scenario where you take the same exact ES cell and you're treating it with different things, so fibroblast growth factor and platelet-derived growth factor, so additional paracrine factors, and then that makes them into glial stem cells, and those could be transplanted into mice as well. So one thing you would need to know is how do we get the cells to go down the pathway, and then how do we introduce them into the organism itself. So embryonic stem cells were all the rage until about 2006 and beyond, when it was discovered that we could make something called induced pluripotent stem cells. And what this involved was surprisingly, to my mind, surprisingly simple. You could take a differentiated cell, you could grow it in a petri dish, so people often would start out with either fibroblast cells or skin cells, grow these in culture for a while. And then if you can introduce really 4 transcription factors, which are shown here at the top of the slide, these cells then convert to a much more embryonic type of status and become highly pluripotent in terms of the types of daughter cells that they can give rise to. So it seems like if we can just get these 4 key transcription factors to be expressed in the cells, that is what you need to convert these cells from being differentiated cells into being something very similar to an embryonic stem cell. And so this was first done, I think, in mice, but then shortly thereafter it was done for human cells. And so here we are with adult stem cell, the reprogramming factors. You know, and don't ask me why these 4, what's so special about them, but that combination, they work. And you guys will appreciate this. I mean, look at all these cell types, and they're representing all 3 germ layers, mesoderm, ectoderm, and endoderm. So that just goes to show like how highly pluripotent they really are, is they've reverted to some status, you know, something akin to the inner cell mass that still has the ability to give rise to all 3 germ layers. So this is great because number one, we can start with the cell type of the individual. We don't have to just use one of these random human embryonic stem cell lines, so that's an advantage. We can do this in culture without destroying any embryos. So that's good too, because that way we don't even have to wrestle with, is it ethical or not to destroy embryos to get embryonic stem cells to use for therapeutic purposes, which you could really make arguments maybe for or against that, but there's definitely that component to wrestle with. And this just sidesteps that whole issue and goes right into a nice source of stem cells that doesn't involve any embryo destruction. So next question would be, are these induced pluripotent stem cells actually pluripotent enough to give rise to any cell type that we would want? And could they, in fact, give rise to germ cells, sperm or egg, that could then be used to pass down the genetic information into a full organism? And so in this scenario, that looks like it is the case. So these cells, this takes you through that process here where we first started out with some skin fibroblasts. We have transformed in or introduced in these 4 key transcription factors that are now expressed in the cells. There's some selection and establishment of these cell lines and induced pluripotent stem cells. Then we're going to color code these orange to keep track of them. You would introduce these into the blastocele cavity of a mouse blastocyst. So this is a host embryo here. And these introduced cells are going to come over and integrate into the inner cell mass and just become a part of that and contribute to that. So now we're going to get an embryo that continues to develop as a blastocyst and then beyond, that has these 2 sources of cells, one from the original inner cell mass cells and second from these iPS cells that we added in there. Now there comes a point where this needs to be put into a mother so that it can go ahead and form a placenta, because we're not really to the stage of being able to grow a whole mouse in a petri dish, of course. So these are just transplanted over into mice that have been given hormones so they think they're going to be pregnant. And now here comes these blastocysts, and they integrate into the endometrium and start forming a placenta. These things are carried to term and born, and when they grow up, they have this dual contribution of the endogenous inner cell mass cells and these orange cells, which were from the induced pluripotent stem cells. Now, some of these mice may make sperm cells, individual cells, that are derived from an entirely iPS original cell. So if that's the case, they carried in whatever genome the iPS genome had, and they can give rise to an offspring that has that full genome now in every single cell of the organism. Does that make sense? So we get a sperm that was derived from iPS, so the organism also derived from iPS. Yeah.
So was that very last one considered a clone then of the very first mouse skin cell it came from?
So this mouse here is considered that one of its parents, if you will, would have come from an iPS cell, and the other parent came from the egg. So it's not really a clone. So, you do this with mice, and the idea would be that we could create an iPS line for patients as well, for human patients that have some type of disease. So we would start out by getting some kind of, say, skin cells from them. We would go ahead and introduce the reprogramming factors, and there are methods to do this, like using viruses to deliver the genes to the nucleus. We would create the iPS cells, which are self-renewing. We can get as many as we want. We can push them down whatever pathway we want. And now we can even genetically modify these. So if there was, say, some mutant gene in the original human being, once we got the stem cells, we could put in a replacement gene as a wild-type copy. And that would be basically the same exact genome, but now a repaired mutant gene, restored to wild type. And then these could be then reintroduced into the same individual and used to repair the defect on the cellular level. So that you have mainly original cells here, but then a few key wild type cells that have been added in to replace the cells that were missing or damaged or non-functional. Okay, so one kind of model disease to try this out with would be something that's a really clear-cut phenotype that's relatively easy to get to and that we can try to repair in a very simple fashion. So sickle cell anemia basically fit the bill here because this is a single gene mutation. It has to do with the ability of blood cells to make a nice round functional red blood cell as opposed to a sickle-shaped defunct or non-functional blood cell. And so the basic process is exactly what I described, where you're going to start with scalpel, cut off a little bit of mouse tail. It's not pretty, but it doesn't kill the mouse or harm them really. It just gives you a source of cells. You can introduce your factors, get some mouse-derived iPS cell clones, and then use a gene targeting method. So it's basically knock-in or knock-out mice method. So ways to manipulate the DNA within the genome of the mouse cell and correct this sickle cell gene mutation. So we're going to put a wild-type copy in here. And we can do any kind of selection that we want with antibiotics or markers or whatever we need to do to make sure that we can recover this population of cells that now has the repaired version of the sickle cell gene. Then, all they did was to start to differentiate these into embryonic bodies. So they're starting to get along the lines of cells that would contribute to hematopoietic cells. So cells in the blood that give rise to red blood cells. And then these were just delivered back into mice by injecting them into the bloodstream. And those cells survived in the bloodstream and were able to proliferate, and in fact were able to differentiate as functional red blood cells. And there was a pretty good recovery of the anemia that the normal mutant mouse would have experienced, because we now have some functional wild-type blood cells that can correct that. So that was a pretty promising outcome. So if we, in principle, thought about how this could be extended to human diseases, there are a number of possible candidate diseases that might benefit from being able to generate a tissue type of interest and stick it in there. So here's some thoughts. Diabetes involves the loss of certain cells within the pancreas that make insulin. What if we could introduce some cells that now are fine for that capacity? Some of these others have very specific sorts of differentiated cell types that inappropriately die due to the disease condition. And if we could A, generate those cells, and B, get them to the right spot, and C, they could integrate, we have the possibility of now restoring at least some of the function, if not all the function, using a tissue type compatible source of cells. And if this worked out, it could just really revolutionize medicine, because at present we have no possibility to treat many of these diseases at all. Like Parkinson's or even heart disease, there's not a lot you can do for once those regions of infarcted tissue die, that's it. You can have part of the heart that doesn't contract quite normally. So here are some of the caveats or main questions that we're now wrestling with in this field. First, can the iPS cells be differentiated to the appropriate cell type? And that, it really just depends on the cell and the status of different types of investigations. So in some cases, yes. In some cases, we don't know, but it seems to me like this is doable, that we could probably wrestle with this and eventually figure it out how to get these cells to do what we want. Second question, what is the basis of the disease pathogenesis? So that means, do we actually know in the disease what cell type it is that has been damaged or is missing or is non-functional? So for example, for Parkinson's, you would need to know that it's the dopaminergic neurons that is the problem here. We have to understand the cellular basis of why the disease is what it is. So again, depending on the disease, in some cases we know a ton about this, and in other cases it's not that clear. Then, what would be the best response therapy? So like, let's say we have the cells and we know what the problem is, what's the best way to correct this? And maybe it seems obvious, but that's going to take a lot of study as well to really nail that down. And so this is a quote from a recent review I read, and I think the general feeling is that this is a field that's gaining momentum, and we're probably going to hear a lot more about it in the upcoming years. So it may seem like kind of a science fiction sort of thing, but the more people study this, they start to find out some kind of quirky things that happen in nature anyway. And this is one of the things that your book points out here, is that in those humans that have been pregnant, there are some cases where it seems that cells from the fetus actually leave the fetus and can go to other regions within the mother and take up residence there. So the fetus, of course, is in a much earlier state, and these cells are much more embryonic-like, even if they're starting to differentiate into heart cells or whatever. And it's just intriguing if some of those cells might leave and take up residence in the mother's adult heart. And so here are some different examples of this, where these cells have been labeled in a way that allows you to recognize their source as coming from embryos, and then contributions made to cardiomyocytes in the heart. These are the cells that contract in the heart. Smooth muscle cells. These are cells that line vessels, blood vessels, and then also endothelial cells in blood vessels as well. So, kind of weird that that happens, and it's probably not extremely extensive, but at least now we know about it. Then there's another related field here, thinking about taking stem cells and trying to build them not into just individual cell types, but into whole organs. And this is a field known as regenerative medicine, because it's going to involve the kind of the intersection of several different fields. So the biology field that sort of finds these cells and can get them to stay alive and differentiate into what you want. Then we have materials design and synthesis. So this would be more from an engineering perspective. Maybe we need to make some kind of a scaffold or a unit that could house these cells and allow them to grow and differentiate appropriately. And then also thinking about geometry, control, and design. So for example, if we were going to make an artificial heart valve, would we want to make it look exactly like the natural human valve, or is there a different design that we might use that just works better in adults for this property and this age? So there's a lot of intersection here between biomechanics and engineering and molecular biology and genomic science and cell biology that's kind of opening up a whole new area of